All right, we'll go ahead and get started. So thank you so much everyone for joining. My name is Lance. I'm a product specialist here at LabKey. I help labs or organizations evaluate Sample Manager to see if it's the right fit for them. I also help labs get fully onboarded and implemented. Before LabKey, I worked in organ and tissue donation as a lab tech and then an account manager. There, I've worked with a bunch of different LIMS, both homegrown and off the shelf, Excel spreadsheets, and paper records. So I'm really familiar with the pros and cons of each system. This makes me really excited to talk more about Sample Manager because I can see how we can help labs and biobanks coming from these systems and improve their sample management in major ways. So today we'll briefly discuss what to consider when looking at a sample management system, who LabKey is as an organization, and we'll dive into a demo of Sample Manager with features within our larger LIMS product. If you have any questions along the way, please feel free to drop them in the chat or Q&A. Also, let me know what types of samples you work with. That way I can kind of make sure that we're using language you're familiar with. And if you need, we can also use different accessibility for closed captions. Just make sure you click it kind of in the bottom right-hand corner. All right, so we'll go ahead and get started into kind of where we're at. So when you're looking at a new sample management system, you'll want to make sure that you're choosing the right software. This can include understanding your lab structure. Does the sample manager system kind of support that structure? How many users can you use and what types of roles or permissions do you need? Then you can also understand, can this software support the number of sample types you have, allow you to capture all of the data that you need? And how easy is it to use? You can look at different key features like freezer management. Can it track how many aliquots or child samples it has or where it's coming from, like what patient those samples are derived from? Do you need an electronic lab notebook? Does it need to support compliance? Are you using labels? Then you also want to consider what to look for in a vendor. Does that vendor provide customer support? Do they have updates to the software? What are their pricing models? And do they support your growth as you get larger? These are all really important questions to make sure that you like the software, that it works for you, and that you like working with the vendor of that software. So I'm going to go ahead and launch a poll that's asking what key features of sample management is important to you. If you can let me know, we can make sure that we spend more time on these different fields. If there's something else that you're needing, let me know in the chat and we can see if we can go over that. So as you're kind of answering that poll, we'll go ahead and dive into who LabKey is. Who are we as a vendor? So LabKey started in 2003 as part of the Fred Hutch Cancer Research Center here in Seattle, Washington of the United States. Since then, we've partnered with a bunch of different organizations from academia to pharma biotech, government research, and a bunch of biobanks. So we're really experts in managing scientific data. We work very closely with all of our partners, not only to make sure that they have the customer support that we need, but also to make sure that we're growing our software in ways that make sense for our clients. With Sample Manager, we have monthly updates to the system with new features, improvements, making sure we're staying up to date in the ever-evolving world of cybersecurity. So we want to make sure that our system stays up to date and that you like using it. With that, with Sample Manager, we focus our software on three main pillars. We focus on data integrity because at the end of the day, this is a software to store your data. We want to make sure that your data is accurate and that it stays accurate throughout the entire process that you're using it. So this just means that we have different data fields where we're capturing information like dates. If you're entering in a collection date, you should only be able to enter in a date. You shouldn't have free text that can kind of muddy that data because that really impacts your ability to track your data and do different queries and make sense of what you actually have in your lab. This kind of leads into sample traceability. We want to make sure that you can search for those samples in ways that make sense for you. You should never lose one of your samples, whether it's you lose data according to it, you lose where it is in storage. You should look in Sample Manager, see exactly where it is in your freezer, go and pull it. Very easy. But we can also track where it is in the lineage. We know where that sample has come from, whether it be part of a specific study or coming from a specific patient. Then you can also track what child samples it has if you're aliquoting or deriving it. Then we really want our system to be flexible. So today we're going to be looking at a system that's set up more for biobanks with like blood and tissues. Uh, but our system can have as many different sample types as you want. You can name them whatever it is that you need. You can also have any sorts of metadata associated with them. So you can track all of the information that's important to you. So with Sample Manager, we have two different tiers. We have our starter tier and we have our professional tier. Our starter tier has our freezer management, our sample lineage tracking, searching and reporting, and our full audit trail. And our professional tier has our assay data management and electronic lab notebook, workflow management, data partitioning through folders, and you can have access to our APIs. We have these two different tiers to make sure that you're paying for what you actually need, so we can make sure that we're more economical as you grow. We also do have a larger product called LabKey LIMS, which has even more functionality, where it goes more into automation. You have plate management, visualization tools, and some conditional formatting to make sure that as you grow and you need more help in the lab and with your biobank, we have that. And then we also have our LIMS Enterprise that has more integrations, more visualizations, and some developer tools. So a lot of our folks do start with Sample Manager Starter, grow into Professional, grow into LIMS, and then grow into our Enterprise if that's what they need. So we'll really be focusing more on the features in Sample Manager Starter and Professional, but we will be in our LIMS environment. So if there is something that you like that you see that we don't necessarily cover, definitely let us know and we can do more of a custom demo for you that goes into these other products. So looking in at the poll that we had, it looks like everyone wants to know where their samples are with their sample tracking. So we'll definitely dive into that. Then we also have a lot of folks that are interested in our freezer management, workflow management, then test and assay results, and some automatic calculations. So we'll make sure we dive into all of these today. But if you have any questions as we kind of go into everything, please let me know. And then once we're done with this demo, we can definitely book a personalized demo to really get more in depth into your specific use case. And we can also help onboard you very quickly should you sign on with us. We help onboard clients in 6 weeks or less. And we work very closely throughout that process and make sure we import your historical data and provide any training that you may need. So with that, let's go ahead and dive into our LabKey LIMS or our sample manager. So on this page, this is kind of our landing page. We have a basic overview of everything that's going on in our lab. We can see all of the different sample types that we have. We have CSF, DNA, nasal swab, serum, tissue, urine, whole blood. These are just names for our sample types. You can really customize these and name them however it is that you need. We also can see what the different statuses for these samples are, if they're available, if they're being processed. These statuses here are also configurable to match the language that you use in the lab. When I scroll down, I can see all of the different boxes I was recently in. So I can see I have 4 whole blood samples in box 21, and I have 77 spaces available. And down here, I have more tissue boxes. We also have our workflow jobs that have been assigned to us. So I can see I have 2 jobs pending, one of which is very overdue. So I should probably get going on that task pretty soon. I also have a list of my recently opened electronic lab notebooks so I can get back to the notes I was taking, whether it's replacing paper forms, keeping track of actual experiments that we're doing, and whatnot. We can collaborate on those notes here. To navigate through our LIMS or our sample manager, we have this dashboard, and this allows me to see all of my different samples and the different types associated with it. So we have data called source types, which is where our samples are coming from. It can be our participants, protocols, patients, specific studies. So this helps us kind of organize where our samples are coming from. We have our sample types, which is the physical samples that we're working with in the lab. It's a bit more straightforward. We have our assays or the tests we're running on our samples and our different storage units. We also partition our data through folders. So this is how we can kind of separate groups of people who may not need access to all of the samples. So if I'm a user in our demo folder, I don't necessarily have access to our P006 study 6 folder unless I'm granted access to that and able to see it. So this is how we can kind of make sure we have some more security with our samples. So right now, let's go ahead and just work in our demo folder. I'm going to go ahead and look at my serum samples. So when working with these samples, it's going to bring up this grid view here. So I can see how these different samples have all of its metadata. All of these different columns are sets of data that I've said are important for my samples. We can configure these to capture whatever data that you need, and we can have as many as you need. All of these can also be organized and filtered through. So if I want to see all of my samples that are currently available, I can quickly filter through that and it's going to show me all my samples that meet that criteria. I can then scroll over, see where they are in storage to quickly get to where those samples are. If I want, I can also search for these samples kind of by searching through their IDs or looking for keywords. From these grid views, I can select whichever samples I want to work on. I can edit them, derive them to have child samples. I can add them to a test, add them to a workflow job, add them to storage, and do a lot more. Whenever you see these grids, you can also download them into a CSV, Excel, or TSV file. That way, if you need to share them with someone outside of your organization, like if you're shipping out some tissues, you can quickly download that electronic manifest. So right now, let's go ahead and click on one of my samples. When I do that, it's going to show me exactly where it is in storage. I have all of its metadata information, but I can also click this lineage tab and see how it's related to everything else in my lab. So for this sample, I can see that it was aliquoted from this specific sample here, and that this sample is part of this kind of patient demo, and it's part of this specific study. I can click on anywhere in this lineage view to also show how it's related to everything else in the laboratory. So I can see that there are actually 3 aliquots of this parent sample. From here, I can also click on the assays tab, see how many different test results we have. So from this parent sample, we can see that one of its aliquots was tested 3 times, get a basic understanding of what's going on. Within Sample Manager, we also have a sample-centric audit log. So this is going to show everything that's happened to these samples from the moment we registered it all the way to when we consume it. So we can see who did it, when they did it, and what they've input here in Sample Manager or any changes that they've made. So we keep very close track of everything that's happening to further help support your compliance. From here, I can also kind of go ahead and click on these. I can see how many aliquots there are. So we'll go ahead and go back to that child sample that we were working on just a moment ago. And let's go ahead and see where it is in storage. So I'm just going to go ahead and click on the exact location it was in. This is going to highlight the exact box that the sample was located in. So it's going to highlight the exact row and column. I can also see whatever other samples are also in this box. Whenever I'm working on these, I can select one sample at a time, or I can do more than one sample to kind of determine how we're working with these. I can then check these samples out of storage. And let's just say I'm going to do this for my demo. I'm checking the sample out. And when I do that, it's going to highlight this space as pink, which means that we're reserving it because we're assuming once you check it out, you're going to check it back in. If I want to clear up this space so another sample can be checked in, I could just move this somewhere like a workbench, or I can remove it from storage altogether. When I'm getting ready to check in my sample, we're going to ask you how much you used when you checked it out. So let's just say I'm using 1/4 of a mil, and I'm going to increment my freeze thaw count. For my reason, I'm just going to say it's for my demo. And if you kind of look back here, you can see we currently have 1/2 mil and our freeze thaw count is 0. But when I check my sample back in, we can see we automatically decrease the amount and we increase the freeze thaw count. So we have a real-time update of what's going on in our boxes and with our samples. That way you're not pulling any samples that don't have anything in it. You can make sure that you're keeping up to date with everything. With these views in our storage, you can configure the boxes to really match the types of boxes you're using. It doesn't have to be a 10 by 10. It can be 9 by 9, 8 by 4, 5 by 3, whatever it is that you're working with. You can also really configure these freezers to match what you have in your environment so they can have as many different shelves, drawers, racks, whatever terminology you're using to make sure that you know exactly where they're going. And let's say your freezer goes offline. Worst case scenario in the lab, very stressful when that happens. We try to make it as stress-free as possible. So when a freezer goes offline, you can move an entire rack and all of its boxes to another freezer to really make sure that you're keeping track of everything that's together, because you're not necessarily wanting to check in one box at a time. You can do a whole batch of them all at once. And then once they're kind of finished, you get the freezer back up, you can move them back very quickly. So from there, let's go ahead and add a couple of samples to a workflow job. So with Sample Manager, we allow you to kind of add jobs to these specific samples, and it's like a step-by-step SOP. So here I'm going to go ahead and do an analysis on these specific samples, and I'm choosing a job template that we've already set up. I can name that job if I'd like. I can add a job owner. I can also notify any specific users that may need to be able to have updates on what's going on with these specific samples. I can set due dates, the priority level, and any attachments or information I may need for the jobs. Usually if you have a shipment job, you're going to be attaching like tracking numbers, maybe in an airway bill to keep track of where those samples are. I can double check the tasks that are going to be going on with these specific samples, assign specific users. So if one user is able to run assays and maybe another one's not, you can make sure the right people get assigned to it. You can also do groups of people. So if a certain department needs to have access to this, they all get a notification that they have a job pending. I can then search for more samples to add to this workflow job if I'd like. But for right now, let's go ahead and finish creating this job. When I finish creating a job, an email will be sent out to those who have been assigned to it. They'll then also see that they have a job pending on their homepage of Sample Manager. Here, I can see all of the different tasks that need to be completed. I can also go to this task page, update who the owners are, and just mark them off as complete as I go through them. So I've completed adding samples to a test. I'm going to go ahead and run the assay. And then once I've added that assay, I can go ahead and import that run. When we import that run, we're able to assign any test results we need to those specific samples, and we're going to automatically import them. Now you can import these samples either through manual input within Sample Manager through the UI or user interface. You can import them through an API, so we're communicating with another system, or you can upload them through an Excel spreadsheet that you import from a data file. So we try to make it as easy as possible. We also have run details, which is going to be the same across all of your samples. So usually it's something like the instrument you're using or the test kit. So if maybe something goes wrong with that test kit, it gets recalled, you can go back and see which samples were impacted. You can also then input the test results for each sample individually, so you can make sure that you're keeping track of what all of those test results are. So we can go ahead and import that and then mark it off as complete. And that's kind of what our workflow jobs look like within Sample Manager. They can be as specific or as generalized as you need to make sure that folks understand what they should be doing within Sample Manager. Another wonderful part of Sample Manager is that once you have all of that information input, we make it really easy to search for samples that meet that criteria. And one of our more powerful tools is Sample Finder. And with this, I can search for samples based off of specific properties. I can search for information based off of a specific patient. So if I want to see all of my samples that came from a patient that was deconsented, because maybe we need to purge that information, I'm going to go ahead and select that and find all of that samples that came from a patient that has deconsented, get rid of that information, make sure we're following compliance. But I can also search for samples that have specific test results within a certain range. So maybe I need to test everything or look for all my samples that tested positive for a certain disease, so we can make sure that we're going to be following our protocols. You can save those searches if you're going to be doing them a lot. So here I can see all of the samples that were created in the last 7 days, go through and audit them. So we do make it very easy and very powerful to kind of work through all of those samples. Another really cool part of Sample Manager is that we also have calculated columns. And our calculated columns can allow us to calculate when we need to be processing our certain samples based off of that collection date. We can search for expirations, but we can also do quality checks. So if I selected a piece of data that doesn't match with everything else that I'm telling it, so let's say I have a plasma tube, I shouldn't be saying that this is coming from a serum type tube. We can mark an error so you can kind of flag it. And then within LIMS, we also have conditioning formats, so we can really make these flags very evident. So here we can see that if my processing window is greater than 2 days, I need to highlight it so we know that we shouldn't be using it. We can also see that in our participants tab. So we're really making sure that we know which patients have deconsented, which ones have consented in that green text, and then which ones are red that are awaiting consent. So it's much more apparent, a lot harder to make any errors that you may in the lab. And then another thing that I'd love to cover are our ELNs. And our ELNs are a great way to not only keep track of your experimental notes and collaborate with everyone in the lab, but it's also fantastic to help replace any paper records that you may have, because paper records are really easy to lose or to ruin, or, scientists don't necessarily have the neatest handwriting, so it also helps with that. So here is kind of an audit checklist that I'm using to keep track of everything that we're doing within the lab, help ensure that we're keeping track of any risks that we may have. Maybe we're not complying with local or national regulations and we need to make changes of that. We can mark that here. We can also tag specific assays, workflows, using that @ sign. So it's a lot easier to kind of search for samples with specific names. These will all then be integrated with that specific sample, so you can find more information of what's going on with that sample. And that sample will also indicate which ELNs it's a part of. So everything is really heavily integrated with each other. So these are really the basics of Sample Manager, and I think it's a fantastic way to really help improve your workflow functions, make sure that you have deeper understanding of what's going on in the lab, and really keep track of where everything is with those kind of tracking purposes. If you have any questions, please feel free to drop them in the chat. I'm more than happy to kind of cover them. I also way more than happy to kind of schedule a personalized demo so we can really dive into your use case. We do also offer guided trials. So if you think Sample Manager or LIMS would be a great fit for you, we can help import a portion of your data, get you kind of going with that trial before you purchase to make sure it's the right fit. So with that, thank you so much. If you want more information, please feel free to go to our website where we have tours. Other than that, I'll open the floor to any questions. And so we can kind of go from there. But thank you so much for joining. So some questions that we usually get is how is the data kind of stored within Sample Manager and what are the IT burdens behind it? So a Sample Manager, it is all hosted in the cloud. We maintain it through monthly updates with both maintenance and new features. So we really heavily reduce any of that IT burden of having to maintain that system. So we can really make sure that you don't really struggle with any of that, and it can help reduce any overhead that you have there. All of the data is kind of there forever. If so long as you kind of keep out your contract, should you end your contract with us, we will export that data and send it to you. We don't hold it hostage. And it looks like we're dropping some links in the chat. If you'd like to book a demo with me, feel free to do that. You can also take a tour for how Sample Manager really helps biobanks. So it is a more biobank specific tour, but of course, feel free to click the link to kind of go to our Sample Manager software so you can have more information there. How easy is it to configure different freezer layouts, particularly LN2 vessels? That is a great question. So all of these freezers, we can kind of just do a very basic one, all have different layouts. So we can have as many different racks with boxes inside of them. These are all really what I tell it to do. So we can just create a freezer really quickly. You can name it whatever it is that you want, and then you can add a storage hierarchy. So I'm just going to do a bulk add of what my shelf one looks like. I'm going to say it's a shelf. And let's say each shelf has 5 racks inside of it. And then each rack will have 4. Let's just go ahead and say 5 by 5 boxes. They can be whatever it is that you want. You can even get more detailed if you want by adding more of them. But by doing that, I have all of these different racks, all of these different boxes. And if all my shelves look pretty much the same, I'm just going to copy and paste it. And it's going to copy all of those different racks. I can rename these to be whatever it is that I want. Doesn't have to be that terminology for shelves, but we really help make sure. And then we can also configure the sizes of boxes. Then you'll just finish creating that. If you have the same kind of freezer layout across all of your different freezers, you can also just copy in the storage definition. It'll create the same exact layout for that specific freezer. We also support labels. So if you have specific labels on your boxes, you can kind of enter that in so you can quickly search for those boxes as well. But hopefully that answers that question. And I should say configuring everything in Sample Manager is also very easy. We do help get you kind of set up and running at first, but it's also easy to do it on your own. You can have as many different labels or kind of sets of data that you need as well. All right. If you have any other questions, I'll stay on for a couple more minutes, but thank you again for joining. We'll be able to send out a recording after this meeting. Otherwise, also feel free to send out a demo request and we'll kind of speak one-on-one too. How would one enter clinical data? Yeah, so there's a couple of different ways we can do that. So if you want to have kind of clinical data on a participant side, you can definitely do that. That's going to be with our sources. So with these participants, you can see that we have like our ages, sex, ethnicity, medical conditions. If you want to kind of organize it in this way, you can definitely do that. You can also have specific visits set up for a patient so you can track them in that way. That one might be another source type that you can use. If you also want it to be under like assays, we could do that. We do have a clinical data system too, so that can also help if you want to kind of track that. I believe that is underneath our LabKey EDC, if that's more in line with what you're needing for more clinical trials and whatnot. We have that, but we can work very closely with you too to see how we can get it to work with Sample Manager. So that's a fantastic question. Any other questions that I can help answer today? And also feel free to kind of email me. I'll go ahead and drop my email in the chat if you'd like to communicate that way. It's just lancel@labkey.com. So feel free to email me there. You'll see my cat picture kind of pop up next to the email. All right. Well, thank you again so much for joining. Always happy to help out and hope you can join us for a couple of our webinars coming up. You can go to our resources, webinars. We have some great ones coming up. Navigating funding cuts, preserving research continuity, and then supporting ISPR best practices using LabKey. If you're at ISPR, please feel free to stop by our booth and say hi. We'll have Hannah and Varun there. It's all the data in a linear fashion. So I think we do have kind of data going from top to bottom, if that's what you're meaning. So we can kind of show how this looks like, where basically the top of our data is stored here. Then we kind of have these different levels and have different tabs for different information organized differently than top to bottom. I would like to kind of speak with you, Jan, on that and kind of what you're envisioning. We can kind of have different sets of data that go like this. Typically, it's going to be streamlined like that, but we could probably work with you in understanding kind of what your specific needs are for that so we can make sure that we're capturing data that you need. All right. But any other questions, feel free to email us. Otherwise, have a great rest of your day. Thank you so much.